Hello again. We will continue with Filippos Tourlomousis. He's an MIT scientist who is doing some very interesting things in Greece. And I will not get into it more deeply, and I will give him the floor so he can explain everything. Filippos?
Yeah, thank you very much. Thank you. I'm doing interesting stuff, I guess, in Greece because interesting things are happening in Greece the last couple of years. And I really want to thank Irini for inviting me here. You know, I'm doing research most of my time, and 80% what we do leads to failures. So Irini gives me this optimism every time I see her, and for the first time, I can announce to her something really interesting that happened in Greece a couple of days ago. But long story short, I'm a mechanical engineer, studied here in Greece, University of Patras, if you know this kind of city, and then moved to the States for 10 years. And the last 5 years, I've been working at the Center for Bits and Atoms, which is the sister lab of the Media Lab, a very interesting place, a unique place, very interdisciplinary, that got funded by the National Science Foundation in the States with $40 million, and really played an important role for all the other logos that you see on the right. And the last year, I came to Greece, and I lead a research group there with funding from the Greek government and the European Union and Resilience and Recovery Fund. And we are doing research in advanced materials and manufacturing. And it's applied research, basically, following the model of the Media Lab, if you're aware about, where we do research that, leads to direct societal and economic impact. And I will try to explain, and I will conclude my presentation talking about our first spin-off called Biological Lattice Industries, where we aim to basically create new materials that, heal and don't treat symptoms. So yesterday, 2 days ago, we announced a really generous grant from the EU RRF, Greece 2.0, 6.4 million euros, and we are building a state-of-the-art manufacturing and metrology facility, basically a place where you can go and make whatever you can imagine. Imagine under one roof all the machines that you can use in order to process and characterize different sorts of materials. And really we want to make this place to be inventive by its heart. And there are specific milestones in the project, and the first one is, what can we do if we have all these machines under one roof, the same way like the Media Lab? And we thought that why don't we empower the academic community in Greece that suffers from underfunding and create open source scientific instrumentation? Really at the starting point when they want to test ideas, help them innovate and not without having access to too much capital. And then the second part is, how do we automate material science? Because material science is a very difficult process. In order to discover a material, you need a lot of work, a lot of capital, and with automation blended with machine learning, something that we call it with this fancy word AI, we really can accelerate this process. So a couple of words about CBA. The same principle under one roof, many different machines that enable applications across different scales. So you can see on the lower part at the nanoscale, something that you cannot see with the naked eye. You can do biology, basically. You can work with the DNA. And if you walk up the scales, you can arrive at very interesting ideas and applications that span, the energy sector, the circular economy sector, and also even the aerospace sector. This is very interesting because I remember my first day at MIT, where my first assignment was to make my own furniture. And I really got shocked about this because after 5 years of PhD, I arrive at the office on the upper left-hand side, and the professor tells me, we have old desks, we need to replace them, I'll cover the expenses to buy the stock material, but you have to make them by yourself. And it was embarrassing because I couldn't know how to make it. So I spent a year learning and playing with machines in order to create and build stuff. And this is the nature of MIT, and this is why you see this tremendous amount of companies coming out of this university because people are able to prototype at a very early stage without having access to too much capital. So I spent a year learning how to cut furniture, basically. And the next year, inspired by my own research, I built the same system that you saw in the previous slide that I used in order to make furniture. This time I used it to make living structures, just for fun. So you see again a 3-axis robot, and in this case, instead of cutting and removing material, we are depositing material that we blended with bacteria that fluoresce when you pass a specific light. And you can see it, you can see those structures. You know, was this a research goal? No, it was fun. It was playing with students there, but we spent couple of weeks trying to understand all the manufacturing process challenges. And actually those learning moments is what led to the creation of my own startup. And this is how Biological Lattice Industries got born. And what you see here, you see again, we are using again this type of robots. Instead of cutting and removing material, now we are depositing again, but also we are applying an electrical field. And we are able to increase the resolution of conventional and commercial manufacturing 3D printing processes by 10x. You can see we can bring down to the 10 micron scale that, you know, even at this point, no other commercial system can do it. So you can imagine a single hair diameter has a value of 100 microns. And we are able to precisely pattern 3-dimensional structures down at the 10 microns, really at the single cell level. So this generates very interesting applications, both for biology research, and especially now with the FDA Modernization Act, and I will explain what is this about, opens a huge market at the order of 1 billion, and then you can use also those structures to guide innate tissue regeneration and healing traumas. So on the upper right side, you saw the system, and this is the first prototype of the machine. And I want to really stress out that we worked with minimum amount of capital, with $50,000 for the E14 fund, just an angel investment, and in a small room with 2 machines. And we're able to prototype and industrialize the scale, really bringing down the cost of the machine so that we are able to scale out and span out the whole factory with minimum amount of money. And this is what enabled, you know, the creation of this startup and trying to close now its seed round. Okay, so this is also, you know, in this way I want to express also the goals of the Greek government project. We want to make the Greek scientists, enable them to be able to do this kind of stuff. So at the Center for Bits and Atoms, you see this new research model, and I call it the research accelerator model because you are not lost in time trying to write academic papers. You are trying to basically prototype, and this is why this lab has generated billions and billions of dollars in companies that now have become even public. You see on the upper hand side, the spin-off from the lab, Formlabs. It's a 3D printing company, a couple of billions in market cap right now, and the company ThinkMagic that created the RFID system that everyone is using right now for telecommunications, but also even the e-ink, all these electronic displays, the electronic paper just came out from this lab just by playing and doing curiosity-driven research and prototyping. The most interesting thing is that, it doesn't stop there. It also gives back to people. And, when this lab started, there was this huge desire from governments around the world to set up fabrication labs. And, if you heard about maker spaces, places where you can go and create stuff and invent stuff around the world, there are almost 2,500 labs around the world that we have set them up in collaboration with the Fab Foundation, a non-profit that runs above the lab, and we are trying to enable people across the world to do the same things. In our pipeline, you can see examples of instruments. A startup company needs, capital in order to create devices in prototype. Especially if you create materials, you need all sorts of equipment, at the order of hundreds of thousands of dollars. And we plan to create an open source, digital inventory for all the scientists so that they're able, with detailed blueprint designs and built instructions. Basically, they're going to be able to download all the instructions to create whatever machine they want in order to do the R&D phase. So, what can you do? Basically, in the lab, we plan to create new materials that gain this functionality by their geometry. And this is very important. You can create new materials simply by manipulating their form. On the lower left part, you see those structures that I mentioned before, those fibrous structures that I'm going to do a deep dive in a bit. And then as you move up, you can even create, recyclable, reconfigurable, modular structures, lightweight structures that have strength similar to steel. So, and this I want to call, related with the previous, panel talking about the circular economy. Very interesting stuff, opening new markets and driving new industries, both at the healthcare level, the circular economy, sustainability, and the energy level. So, what is the main problem in order to be able to innovate and create devices and materials? I think it's reflected in this slide. On the left part, you see Thomas Edison. We all know that did thousands of experiments in order to discover the incandescent lamp, but electrical bulb. But also on the right side, you see grandmother, creating this cake. And it's very interesting because no one knows how to replicate this recipe. Even if you move here and put her to another kitchen, she cannot have the same performance. And what this reflects, it reflects the current material science paradigm and all the, the fact that it's manual, trial and error, time-consuming, and expensive. And this is the problem that we're trying to solve with automation and machine learning. Systems that are driven by those two aspects. So let's move and look at the healthcare level. What are the problems that we have right now? We don't want another Me-Too product. And what I mean by that, we don't want another stent, we don't want another catheter, we don't want another pill. Because what happens, the reality is that medicine suffers the last decades from a very serious problem. Doctors are trained to treat symptoms. They are not trained to heal and eradicate and heal traumas. So there is a market. It's called the combination product market. It's expected to be, $214 billion by 2027, and it's been driven by those products. Now with tissue engineering and regenerative medicine, you can use materials that basically, give all their functionality from the geometry, they biodegrade in the human body, and they guide the innate tissue regeneration. It's literally not an evolutionary step. It's a revolutionary step, I believe, for medicine, and there is a huge opportunity for medical device makers. Okay? So let's talk about biological lattice industries and those devices. It is very well known that those companies need a lot of capital to commercialize, okay? And we've seen the funding and the financing plummeting both across the United States and across Europe. And the problem is that people that create those companies started those companies believing on a biology dogma. And this is the central dogma of biology, that DNA is the CPU. DNA makes RNA, and RNA makes proteins, and proteins give us the functionality. And you have this one-way flow of information, okay? But if we imagine something else, and instead of using those two-dimensional plastic petri dishes that you see biologists culturing cells and trying to create drugs based on these studies that are not replicas of the actual human scenario, which is 3D, soft, and much more complicated. And what if we could reverse this dogma? And instead of programming functionality by changing the DNA, changing the environment around the cells. And you can do that if you are able to create those materials down at the single cell level, because you can change the shape physically, reproducibly, based on a robot and a machine that you're going to program, and you're going to have reproducible biological functionality, which is really revolutionary for biology. Now, this is not coming out of my head. It is very well known that biology suffers from irreproducibility issues. Billions of dollars have been lost from the National Institute of Health, NIH funding, for non-reproducible research. We know that drugs, 90% of them, come at the human clinical trials and then fail because animal studies don't replicate what is happening in the human scenario. And this is what we want to solve. this is an example of a company that started years ago and showed that, if you have access to, infinite capital, hundreds of millions of dollars, then suddenly after 10 years you can say, what I promised to do is not reproducible, and now I have to reconsider all my goals. And Organovo is a very well-known case study. And now, if we can create at lower cost, faster, better therapies using those materials, we could do it in a rational way. And this opens great opportunities, because no more we are going to fall in this valley of death where we need infinite amount of capital in order to commercialize. We believe that the problems here that we are solving is the issues of the cost, the scalability, and the reproducibility in biological sciences, okay? And the key thing that we are thinking when we create those products is we are thinking in terms of regulatory. We think in the lab like being and working with the FDA from day one. So you have to think when you create those devices, something that is called the primary mode of action. Okay? If all the scientists knew from the beginning what the primary mode of action is, then we would have much more innovations, bigger number of innovations right now, because it's very interesting. You can have a primary mode of actions. Basically, you can drive the functionality of your device based on chemical signals. Okay, if you add chemicals, you get the functionality that you want, and then you go from the drug regulatory pathway, hundreds of millions of dollars. But if you create devices that function and get the result based on mechanical signals, on the geometry that we program through our robots, you go through the medical device pathway, and it's much lower, and you can have the same impact. So the mission is, how can we create architected biomaterials that, if we implant them, they can guide the innate regeneration at trauma positions in order to heal, okay? So the goal is here to create perfect mechanical analogs of the tissues that have problem, okay? And I will talk about applications and where we are heading on. So we have a manufacturing system that is able to print structures and have topologies that you can see across the whole dimensional scale window. On the left side, you see nanoscale nanofiber membranes. On the right side, you see low resolution, three-dimensional printing processes. You see from the scale bar, those individual strands are at the 0.5 millimeter. And we come and we fill in this dimensional scale window with a novel process using those electrical fields, and we are able also to print down to the single cell level. Now, one more time how the process works. You have, a material that is FDA approved. We took that, we considered that from the beginning, not a new material, an actual material that has been used in the medical practice for many, many years, okay? And you have it inside a syringe. You heat it up, you melt it, it behaves like honey. So you can push it with air through a small needle tip, a very thin orifice. And right at the orifice, you apply an electrical field. And because it is viscous and elastic, you can stretch it. And you can get down to this, single to 10 micron scale level. And you can print those structures in a very scalable way, in length, width, thickness. And also, you can make them, as I mentioned before, you can make those robots at a very low cost if you want to span out a factory and you want to print them in high volume. So in short, we have a scalable manufacturing process. We are using medical-grade polymers, not weird materials that need years of regulation. No chemical solvents at all, just a pure medical-grade material. And through an interesting software and hardware platform, we are able to create those high-fidelity structures that we hope to use them as therapeutic devices. I'm really happy because this is work that I have been doing the last 10 years. It's like almost 3 years ago then General Electric characterized our process as the 5 coolest things that happen on Earth. MIT News featured it, even The Economist. And we really thought it before we spun out this company. It's almost 2 years now, and we are really excited to put those structures, in the market, both for research consumables to enable biologists to do better biology, but also as therapeutic devices. There is a patent, and actually there is data, and we have demonstrated the following thing. If you get those structures and you simply change the architecture, you change the shape of cells, and really you change their functionality. And we have demonstrated that with stem cells. Stem cells are cells that reside in our body in very small quantities, and they are neutral. They don't know what to be. They are not skin, they are not bone, they are nothing. But they are those magic cells that when you inject them, they have this therapeutic capability, and they start the innate regeneration capacity. So by changing the architecture simply with the robots, we were changing cell shapes, and we were guiding them to become different things. And there is a family of patents, and this patent last year, the first one, got granted at the USPTO. So the business, I think when you have a technology platform that allows you to create materials across different scales, you can program those materials at different architectures to match mechanical properties at different tissues inside the body. You get this huge opportunity of $134.9 billion. It's an opportunity. It's not an addressable market size, but if you demonstrate something in the beginning, this opens new capabilities, okay? The first problem that we're trying to solve is breast cancer. Okay, think about women that have breast cancer. They undergo breast mastectomy. Okay, they go the surgery, the breast mastectomy. The breast surgeon removes the tumor, and then you have plastic surgeons doing the breast tissue reconstruction. But how are they doing it? They are doing it by simply replacing it with fat tissue that they extract from the other parts of the human body. A disgusting process, really replacing tissue. Now, what we are trying to do is to create those structures that fill in those voids, and they guide the innate tissue regeneration and the healing process, and it's like never the problem existed before. This is the goal, okay? So, let's see how we would solve this problem. We have created a software infrastructure, okay? We know from the surgeon that more or less what is the shape of the tumor that we're going to remove. Based on that shape, we run a couple of algorithms, and we are matching the shape with different architectures. And these are digitally made. This is what we are going to print. The next question, the important question, is which one of these architectures corresponds to breast tissue? Because it has to be a perfect replica. Because if you implant it and it's not the same, it will generate foreign body reaction. You're going to start to have inflammation. You're going to start to have necrotic tissue and fibrosis. So the design tool gives us the shape and the architecture. The metrology tool basically talks with the design tool and tries to optimize and tell you, based on the mechanical properties of the host tissue, of the breast tissue, which architecture is the optimum. And then this works, in an iterative, closed-loop manner, purely digitally. Can you imagine if you had to print, test, do experiments, how much money you would have lost in the R&D phase? Now you can do it end-to-end, with simulations, the design tool that gives you the architectures, runs the simulations, tells you what is the mechanical response, basically if it's close to the breast tissue, and then there is a master tool that is not those fancy deep learning algorithms and chat GPT that we hear every day. It's simple statistics that does decision making. It tells you where to search in this huge design space in order to find the optimum product, okay? You can see the manufacturing process, live here. We have online process control because what we print, we want to know exactly what it is. You need to do quality control in order to industrialize it. So we have deployed computer vision algorithms that people call it AI. So we recognize where the fiber is, we measure it while it comes out from the needle, when it is deposited, and we get this huge big data set. And at the end of the day, we can give it to the FDA and say what we promised to print, this is, happening. And then this is the whole technology platform, and we call it the AI-driven foundry platform. Because if it works for breast, and we have all these different capabilities to print, then it's going to work for bone, and it's going to work for cartilage, and opens these huge, market opportunities. So for breast, I think it's the right moment for breast because there are a couple of companies around the world, not too many, actually, that are trying to enter the clinical trials. They have different manufacturing processes, but if you see the amount of money that they have raised, this it's not friendly for the investors. So we believe with our technology, we have decreased dramatically the amount of R&D that is being needed, and we hope the next 2 years to identify which one are the optimum products for breast, bone, and cartilage, and after 2 years to come out to the market and start selling all around the world scaffolds that are perfect replicas of the human in vivo scenario and use them as research consumables without having to do any regulatory pathway. That's all from me, and I'm happy to get any questions that you might have. Feel free to ask whatever. I tried to keep it simple, but it always becomes technical, you know. Yeah, sure. Oh, cool.
Thank you for the interesting talk. I was just wondering if you seed the mesenchymal cells prior to printing, or whether you inoculate the lattice after printing.
Yeah, so the results that I showed, and they are covered with IP, is we expand them for 1 week on T75 flasks, and then without any, bioactive molecules, just the pure food, fetal bovine serum, and we seed them then on top from the scaffolds, and the scaffolds are not chemically functionalized like with fibronectin or something like that. They just, migrate and take shapes and give you the functionality afterwards.
That's fascinating, thank you, because I was imagining them being printed and just the shear forces of being printed and perhaps the temperature as well.
That's exactly the problem of 3D bioprinting, putting cells inside syringes and, undergoing this stress and then trying to recover. That's why I believe 3D bioprinting is going to fail.
Thank you so much.
You're welcome. Great question.
Yes, my name is Gleb, Relay Capital. So I don't have a degree from MIT, so probably my question would be a bit more naive. So you're saying that you're replicating the cell structure of different cell types.
Of different tissue
types. So do you customize this for every individual patient, or there is some sort of generic pattern that you've discovered for every single tissue type?
No, that's a very important question. Okay. So, related to patient-specific stuff, okay, you can, we can make it patient-specific based, on the tumor, for example, for breast, the shape. But there is a huge variation across different ages for different tissue types. So we are working with surgeons in order to make them patient-specific across age groups and for specific tissues. So we cannot get, the tissue of one patient, test it and try to make it replica, because this wouldn't support any economy of scale and commercialize it. But there is a range, and we believe for specific tissues and different ages, age groups, there is universal architectures for that. I don't know if I answered your questions. We can discuss more if you want afterwards.
So we'd like to thank Filippos for coming. And
Thank you.
He will be available for any. Thank you again.
Thank you so much.